Now I'll show you the results we also had on lichens. As Alice also told you, you already know the method and the context, but just to remember you, in this case we're using ecological indicators, lichens, and we want to see lichen functional diversity along a gradient in space, because we believe that that gradient in space can be used as a proxy of what is going to happen in time. And we hopefully with this we will be able to build the early warning indicator. So to do this, we focus, but just to begin, I wanted to show you the traits we have chosen. So we focused to this work that I'm going to show you only on 3 traits. So the type of photobiont, like I said, lichen is a symbiosis, so it can, within that symbiosis can be, it's between a fungi and a green algae. Cyanobacteria, or a special kind of green algae, which is the Trentipolia, the one that I showed you yesterday, which is kind of orange. We also focus on growth form. So the 3 main growth forms: crustos, if it's like a crust; folios, with the leaf-like form; or fruticose, which is more like a shrubby 3D dimensional thing. And the type of reproduction. Lichen can reproduce both sexually and asexually. So in this case, we divided them in sexually with apothecia, or asexually by Ceridia, which is a kind of more like lichens produce Ceridia, which are more or less like flour or sugar above the surface that can disperse with wind, or isidiate, which is a kind of more complex in structure, and usually breaks with water and goes with the water to disperse. This is just to contextualize, because these are the traits and functional groups we are going to see next. So, first of all, because I didn't explain you this on Monday, the context of all this work within a microclimate, and in this case an aridity gradient, was done because of a previous work of Pedro. What he did, and just to show you how good they are as indicators of climate conditions, he did a work in a place north from here, from Lisbon. And what he did, this is a really small area in a forest. And what he did is sampled lichens along in all these sampling points. And you have been hearing a lot these days. He's talking about potential solar radiation, which is a kind of, and Pedro, correct me if I'm wrong, an environmental variable which takes into account the energy, gives you the energy on a sampling point. So it takes into account the orography and the incident light on that point, taking into account also the surrounding orography. So imagine a place here, which is north, won't receive energy. Even the amount of energy that it receives cannot be the same as a place standing on a south-facing slope of the mountain, okay? So what we did is sampled lichens on this mountain in Serra d'Aire, and he wanted to see, and this is the values for potential solar radiation, and he wanted to see if you could see, regarding the photobiont type as a trait, if you could see some changes, what lichens would show us. And what he saw was that, in fact, if you plot it against potential solar radiation, they would behave, green lichens and cyanobacteria lichens would behave completely different. So cyanolichens, to be active, to produce, to make photosynthesis, they need liquid water. So they usually appear in really wet sites because they don't stand too much drier conditions, okay? While green algae lichens, they are able to activate only with water vapor. So they can withstand drier conditions than the other ones. And what we see here is exactly that. So you can see that green algae lichens, they increase with potential solar radiation, so as the amount of energy going to that place increases, while the others are not able to do it. So if you map the LDV, remember the index that we measured, we learned how to measure. So if you map them in that space, these two functional groups would show you that conditions, the conditions of the energy arriving to that place. So the microclimatic conditions in this case, because the microclimate is the same, okay? So we are talking about a small space in a small area. So when we saw this, we thought maybe it will also work if we have a long and gradient in space. So in this case, remember, like Elise told you, we have our aridity gradient in the south, and I completed this gradient enlarging it to Spain. So we sampled in all these, so this is where we were yesterday, okay? And the most arid part in the Iberian Peninsula is in Spain. So we enlarged the gradient, and I did some more sampling sites in Spain to enlarge the picture. And what we saw, in this case, we wanted to do, and this is just, these are the first results because we still haven't looked at everything. So in this case, what we wanted to do, and this is a non-metric multidimensional scaling, so we wanted to see if plotting the species abundance and the functional groups in that gradient, we could get a picture of what are the relevant traits that are responding to the aridity gradient. And remember, like Elise said, in the case of lichens, epiphytic lichens, with Laura is a different thing. We are using response traits, okay? So they don't have in our ecosystems a really prominent paper role, okay, on the ecosystem, but they respond quite clearly to everything that's happening on the surroundings. So what we wanted to see here is, in this gradient, do we have traits that are responding to aridity or traits that are more related to the more wetter parts? And in fact, we did. So what we did find is that in the less arid places, there is a predominance in these traits, in these functional groups. So crustose species, fruticose, remember the shrubby ones, and lichens with trentipolia, meaning the more tropical ones that need more humidity, appear in the less arid places. And these places correspond to the places in Portugal, okay? And this is not so surprising because Portugal, you know, we have the Atlantic, so we are quite more humid than the continental Spain. We receive the winds from the Atlantic, so air is usually more humid, and so we get much more humid conditions. And on the other side, we got the traits related to the most arid parts of the peninsula, okay? So we get the foliose lichens, the 2D lichens, the dry-resistant cyanolichens. And remember the work I showed you before? We had the cyanolichens responding in the most wet places. But in cyanolichens, we have two completely different kinds. So the ones we call the emblematic, which appear in the northern hemisphere or in the wet temperate regions that need lots of water, and those are usually big. And then you have the conspicuous species, which usually are really dark, black, and that you can find them in the deserts, for instance. And those are the resistant cyanos. So the cyanobacteria, the cyanolichens I have here, are the dark cyano, okay? The small ones, dark colored, usually black. And isidiate species, so the species that reproduce by isidium. And this is, well, this is kind of, this result, the fact that we have cyanolichens there, it's quite interesting because usually everybody says that cyanolichens are in wet parts. So this means that this is important in terms of ecology. So imagine if you mix them all together, you won't see this picture because within the same trait, they are responding in completely different ways, okay? And the other ones, well, it's quite, this pattern of foliose was also seen in some other places, like the UK. Foliose are usually appear in more continental places, so with the less humid conditions because of the structure. They are they can absorb better the water, so it's normal that they are more efficient in these places. So after having this, the next step would be, because we want to do we want to see we want to build the early warning indicator. So what we did here, and I'm sorry, this is so small. It's more or less what Elise did. So in this case, I picked up the scores of the NMS. Remember, the first axis of the NMS is the aridity gradient. So we picked up the scores and we plotted against the global aridity index to see if we have the same pattern as in plants, okay? If we can spot where the transition occurs, and with that later build the early warning indicator. And in fact, we have a nonlinear relationship. The thing is, here we know where the transition is occurring between the two ecotones. So in this case, we sampled the semi-arid. And this is the ecotone between semi-arid and dry subhumid, so the less arid ecosystem after this one. But of course, in this sampling I showed you, we stopped at 0.5, which is the boundary between the two ecosystems. So the thing is to build the early warning indicator is what will be the trend after. This is the aridity index of the region, okay, the Iberian Peninsula. And if I map this is what you get. So the idea, just to show you what we want to do after, because we need more, and with that I can tell you this. The thing is, we don't know if the trend would be this, okay? If you have an increased variability that, like they have in plants, and then in the next, in the dry subhumid, this will happen, so then you could build, maybe the early warning indicator will be somewhere like here. Or if you continue sampling and doing it like that, and probably the ecotone is bigger, or they are giving a different signal. So this thing we still don't know, because what we want to see is the early warning. So, doing this, what we want to see next is, if you pick up this, the results of lichens, and you see, this is the boundary that lichen says that it's occurring already, the transition. So if we map it, you can see that it's a little bit earlier than what, for instance, the aridity index says, because the aridity index says that, okay, here at 0.5 is where the dry subhumid starts. But lichens are already seeing a transition, or you're already seeing a transition before. Okay, so the idea, just to show you how after we will build the indicator, is to see this. So knowing this, we can then tell you, okay, something is already happening, even if you cannot see it, for instance, on plants or whatever. But we know that something is happening here. So having this, we can already map and tell you areas that probably are already in transition. And this would be the final picture. And because we want to apply this and we are using functional diversity, the idea was to do, like Elise said, the universalization. So this model, after built, has to be tested somewhere. So we also did some work in Thailand, Brazil, and the United States, and we want to see exactly the same thing there. I don't have the results already treated, but I can show you that, and this is just, the colors are different functional groups, so the ones that I was showing you, and this one's just related to growth form. And each of these is a different transition in different areas of the regions of the world, just USA is not included here. And you can see that they shift completely, so probably we will see exactly the same transition, and even if you are, this is just to show that even if you are in a different ecosystem, if you use the functional approach, you can still build the indicator and it will give you the same answers. So, thank you, and if you want to join us and try this in your country and try to see if the model also works there, we will be glad to help you and collaborate with you. Thank you.